A tumor microenvironment is a densely organized ecosystem of interacting cells. To sequence the RNA using bulk methods, you must homogenize it. This captures an average profile of gene expression, but it destroys the cellular heterogeneity required to see how the tumor is actually organized. Single-cell RNA sequencing originally solved this masking effect by isolating and profiling individual cells, examining them one by one. However, that isolation requires enzymatic dissociation. You pull the cells apart, and in doing so, you permanently strip away the physical coordinates of where each cell existed in the tissue. To map the mechanics of a disease, knowing what transcripts are present is insufficient. We need to know exactly where those transcripts are located relative to each other. Spatial transcriptomics is the revolutionary new technology that captures detailed gene expression while perfectly preserving the intact tissue's physical architecture. It allows researchers to map high-dimensional molecular data directly onto traditional two-dimensional histological coordinates. The field is defined by two mechanically distinct approaches: sequencing-based spatial capture arrays and also imaging-based in situ decoding. Selecting between these two methodologies is the defining architectural decision for any modern pathology or clinical research pipeline. Next-generation sequencing, or NGS-based spatial methods, rely on solid-phase microarray capture. A standard 10x genomics Visium slide features a 6.5 by 6.5 millimeter capture area, containing 5,000 distinct circular spots. This diagram shows a single 55 micrometer spot, containing millions of immobilized primers. Each features a poly-T tail, a spatial barcode, and a unique molecular identifier. To capture mRNA, the overlying tissue is permeabilized, releasing strands that diffuse downward and perfectly bind to the primer's poly-T tails. Because the poly-T tail catches any messenger RNA, this mechanism provides unbiased whole transcriptome coverage without needing pre-selected gene panels. The constraint is physical resolution. A 55 micrometer spot captures the combined transcriptome of roughly 1 to 10 cells, meaning it lacks single-cell precision. These microarrays excel at generating broad molecular atlases across entire tissue sections, but they average out the data at the multicellular level. BGI's StereoSeq platform was engineered specifically to close that 55 micrometer resolution gap. Instead of printed microarrays, StereoSeq utilizes patterned arrays constructed from DNA nanoballs, generated through a process called rolling circle amplification. This diagram shows the extreme shift in scale. StereoSeq spots are just 220 nanometers in diameter, separated by 500 nanometers of spacing. This density enables transcript capture at sub-micron resolution across a centimeter-level field of view. While this physical architecture circumvents the resolution limits of standard microarrays, mapping tens of billions of probes introduces massive computational and data storage challenges. The alternative to capture arrays is imaging-based spatial transcriptomics. Platforms like Xenium and Cosmox abandon the solid-phase substrate entirely. They probe RNA molecules directly within the intact fixed tissue matrix itself. This diagram illustrates the padlock probe. These probes hybridize to specific transcripts, circularizing into a closed loop upon matching. Rolling circle amplification then spins out a nanometer-sized knot of repeating DNA anchored to the RNA. Finally, cyclical waves of fluorescent reporters bind to the knot, flash brightly, and are cleaved away. This flashing sequence creates a 64-bit fluorescent barcoding scheme, allowing algorithms to map specific transcripts to their coordinates within the cell. Because these padlock probes must be custom designed for each target, imaging-based platforms are restricted to targeted panels of 1 to 5,000 genes. They cannot offer the unbiased discovery of a microarray. Imaging-based platforms trade the breadth of the whole transcriptome for higher morphological and spatial precision. Beyond the theoretical resolution limits, these platforms must navigate the physical constraints of clinical sample preservation. The formalin-fixed paraffin-embedded, or FFPE tissue block, remains the undisputed global standard for archiving clinical cohorts in pathology departments. The chemical cross-linking required to make an FFPE block severely degrades RNA, breaking the chemistry of many spatial omics protocols. This matrix compares leading platforms by resolution, coverage, and FFPE compatibility. Broad institute benchmarking shows viability sharply diverges. Visium 2.0 and Xenium operate effectively on FFPE samples. while ultra-high-resolution platforms like StereoSeq remain restricted to fresh frozen tissue. The benchmark data demonstrates that Xenium consistently generates higher transcript counts per targeted gene in these degraded FFPE tissues, maintaining accuracy without sacrificing specificity. The optimal platform choice is rarely dictated by pure resolution specifications. It is governed by the physical state and availability of the clinical archive. Successfully running a tissue slide and generating optical data is only the first step. The true bottleneck in spatial omics is post-experimental computation. The hardware outputs raw pixel arrays. It generates raw optical reads and records specific barcode coordinates. These data points have no biological context without spatial boundaries. This requires cell segmentation algorithms. These algorithms analyze DAPI-stained nuclei in the tissue image to draw boundaries around individual cell bodies, capturing specific transcript dots within them. Bioinformatics frameworks, such as Seurat version 5, use these computed borders to assign hundreds of thousands of isolated transcripts to specific individual cells. The output is a structured cell-by-feature matrix. Researchers can query this matrix to map cellular neighborhoods and compute ligand-receptor interactions across the tissue. While the chemical assays capture the raw molecular signals, it is computational geometry that actually defines the cells. The next evolution of spatial bioinformatics moves past simple cell counting, utilizing multimodal foundation models. STORM is a new multimodal foundation model designed to integrate transcriptomic data directly with standard H&E histology images. The model was trained on 1.2 million spatially resolved spots, precisely paired with matched high-resolution H&E images spanning 18 different human organs. This flowchart illustrates STORM's architecture. Dedicated spot encoders independently extract features from both the H&E image patches and the spatial transcriptomic data before merging into a single central spatial encoder. This network is spatially aware. It analyzes a 5x5 neighborhood of spots to compute holistic tissue organization rather than evaluating isolated data points. Because STORM was trained on both modalities simultaneously, it can now infer spatial gene expression and predict patient outcomes from a standard H&E image alone without needing the complex transcriptomic assay. This predictive capability has been validated across 23 independent real-world cohorts comprising over 7,200 patients. Spatial transcriptomics has moved beyond a discovery tool for research atlases. By fusing visual pathology with molecular data, it is transforming static tissue images into predictive models for clinical outcomes.